Oh,
our speaker is not visible.
Afternoon, depending on where you are. So let me introduce the seminar series and its objectives. I am Professor Sujit from IIT Madras. So Critical Transitions and Complex Systems, or CTCS as we call it, seminar series is being jointly hosted by Indian Institute of Technology Madras in India and Potsdam Institute for Climate Impact Research in Germany. This seminar series brings together experts from a variety of fields such as climate science, ecology, neuroscience, fluid mechanics, etc., and aims to disseminate the state of the art in the prediction of critical transitions in these diverse fields of study. The seminar takes place every month on the last Monday of the month, so mark your calendars. Before I introduce the speaker, a few housekeeping notes. I request everyone other than who is speaking to turn off their microphones, and please type your questions in the Q&A box in the Zoom bar so that a speaker will take up these questions at the end of the talk and answer all your questions. We have ample time for questions. So with this, I invite Professor Kurtz to introduce the speaker.
Okay, yeah, thank you very much and welcome all. I'm really very happy to introduce today and to say hello to Professor Ehud Meron. He is from the Ben Gurion University, and you know this area is now in a rather difficult situation, but nevertheless he was willing and is willing to give us a talk, and we all know that he is very active despite all the problems. And his main topic is ecology, but in a very special manner, let me say. One people would call it theoretical ecology, but he called it as a title of one of his books, Nonlinear Physics of Ecology. I think this best describes his approach because he combines in a very original way nonlinear physics, nonlinear dynamics with challenging problems in ecology, especially also these days under climate change. And he is both famous and important. Some people are only famous, some people are only important. He is both. That's really already shows how influential he is. And he has made a lot of really substantial and fundamental contribution to this field, which was very young when he started. Now it's established mainly due to his work. And he has published what is also a typical criterion for people who work interdisciplinary, that he could publish in the best journals in physics, as PRL or Physics Reports or Chaos, of course, but also in leading journals in ecology, ecological modeling, theoretical ecology, and several others, but also in these general journals as PNAS or Nature Plants and several others. And this already shows how well-known he is and how substantial his work is, that it's treated really as important in both fields. And I'm very interested in his new research and his view of ecology, and I think we all can learn a lot from this. And I give the floor to you, Ehud. Thank you very much.
Thank you, Jorgen, for the kind introduction. I'm not sure I deserve everything you said, but it was very pleasing to hear that. Let me share the talk. Do you see the...
Yes, I can see it. You can please make it full screen. Yeah. Very good.
Okay, good. So climate change and the development of dry climates threaten ecosystem functioning and the many services that ecosystems provide to humans. Worldwide, and especially in drylands, water-limited systems that occupy more than 40% of the terrestrial Earth area. In addressing ecosystem functioning, it makes sense to start with the lowest trophic level, the level of plants, because they affect the whole food web. So the response of plant communities to water stress is likely to involve a mechanism operating at different levels of ecological organization, starting with the organism level, the level of a single plant where phenotypic changes can take place. Phenotypic changes are changes in the properties, traits of plants that in response to environmental stress that occur on a relatively short time scale. These are not evolutionary changes. They occur on short time scales, sometimes days, even minutes. And one example relevant to this talk of phenotypic change is the ability of plants to grow roots toward the soil volume that are richer in water, a behavior that is called hydrotropism. At the community level, community level is the level where we have several or many populations of different species. Community reassembly can take place in response to environmental stress in the form of transitions from fast-growing species to stress-tolerant species. One example is a transition from so-called C3 plants to chem plants. The difference lies in the photosynthetic pathway these plants exhibit. C3 plants, which are the majority of plants, like 85% of them, take carbon dioxide through the stomata. The stomata are little microscopic openings in the leaf through which the plant exchanges gases with the atmosphere. So, in particular, plants absorb carbon dioxide through the stomata, but also release water. And C3 plants open the stomata during daytime, and the problem with this behavior is that under water stress, the plant may lose too much water to the air because of the low vapor pressure during daytime. Chem plants open the stomata during dark time, and where the vapor pressure in the atmosphere, the air is higher, so they lose less water, and so they are more stress tolerant. But at the same time, they process the carbon dioxide and proceed with the photosynthetic pathway more slowly because the carbon dioxide cannot process in the dark. It needs products that is produced by the photochemical reaction during the day, and it's short-lived, and therefore it is stored in malic acid in vacuoles. These vacuoles make the leaves very fleshy, like in succulent, and released back the next day. So this slows down the photosynthetic pathway, and this is why chem plants grow more slowly. Yet, a third mechanism to cope with water stress is spatial self-organization or vegetation pattern formation. This occurs at the population and or community level. Population, we're talking of many species, many plants of a given species. Community is several or many populations of different species. And this is an example of a vegetation pattern, stripe pattern in Australia of acacia trees. And the reason why this is a mechanism to cope with water stress is because the surviving trees here benefit from the water in the surrounding bare soil, and they have various mechanisms to draw this water for their benefit. We'll come to that later on. And there are many examples of vegetation patterns, starting with the hexagonal gap pattern, nearly hexagonal gap patterns. This is grass pattern in Australia. Each gap here is about 40 meters in diameter on average. These stripes of acacia tree, also in Australia, spot pattern, spot patterns, labyrinth-like patterns. This may occur with wood vegetation or with grass. In the case of grass, the typical length scale is 10 centimeters. Here it's 100 meters, or 3 orders of magnitude difference, but the patterns are very much the same. And there are more exotic patterns like rings, arcs, isolated spots, localized patterns, and actually multi-scale patterns. We see here small spots within larger spots. So these mechanisms are likely to act in concert for the simple reason that if one mechanism is active first, it relaxes the water stress to some extent and weakens the driving force for the other mechanisms. In this talk, I would like to address the interplay between vegetation patterning and phenotypic changes, and between vegetation patterning and community reassembly. But before that, we need some background in vegetation pattern formation theory. That theory tells us that vegetation patterns are formed by a positive feedback loop between local vegetation growth and water transport toward the growth location. And there are several water transport mechanisms, which I'll describe in a moment. But this general feedback already helps us understand how vegetation patterns can form. So starting with uniform vegetation, green represents biomass of the vegetation. Suppose that by chance, there is a patch of denser vegetation. Then, according to this feedback loop, the plants there will draw more water from their surrounding, more than the surrounding does, so there will be a net flow of water toward the vegetation patch, and this will make it yet denser, but at the same time, this will make the surrounding vegetation sparser. And you see that this is a mechanism that can create patterns. Those of you who are familiar with activator-inhibitor systems may recognize that this is another example of an activator-inhibitor system, where the activator is the vegetation biomass. The vegetation enhances its own growth by reproduction. A plant disperses seeds, which germinate and grow into new plants. But at the same time, it takes up water from the soil and causes water deficiency, which inhibits further vegetation growth. So water deficiency is the inhibitor. And as you might know, in the case of fast inhibitor diffusion, we get stationary patterns. And this is the case here, as the inhibitor, water dynamics, is much faster than the vegetation dynamics. There are several mechanisms of water transport. One of them is overland water flow. This is typically the case where the bare soil is covered by soil crusts. This can be physical crust by the impact of the rain, or biogenic crust, like crust covered by cyanobacteria that need sunlight for photosynthesis. Those crusts reduce the infiltration rate of surface water into the soil. Under vegetation patches, litter, for example, blocks sunlight, and so this biogenic crust cannot develop. Physical crusts also don't develop because of the absence of impact of the rain. So there is infiltration contrast develops, low in bare soil, high in vegetation patches. The root zone also makes the soil more porous, which is increases even further the infiltration rate in the vegetation patch. So altogether, because we have here vegetation edge, it draws the overland water flow to the plant. The vegetation grows even more, sharpens the infiltration contrast, increase the overland water flow, and this is a positive feedback flow. Another mechanism is water conduction by laterally spread roots. And yet, if the roots are local, then the water uptake in the root zone deplete the water there and create soil water gradients and soil water diffusion from the side toward the root zone. Now, this is a model that came up 20 years ago. It captured all those three water transport mechanisms and the feedback loops associated with them. So we have here a system of three partial differential equations, nonlinear equations for the above ground biomass, soil water content, and surface water. I won't go, I don't have time to go into much detail, but I'll just say that the important parameter is precipitation, which increases surface water, but surface water is reducing by infiltration into the soil, and the infiltration rate is biomass dependent to mimic the effect of soil crust. So it's low in bare soil and high in vegetated patches if this parameter F is small. And we have here expression, non-local expression for the biomass growth rate and the water uptake rate. These are non-local because of the lateral spread of the roots. So the biomass growth or vegetation growth at a point X depends on water at a point availability at point X prime, which the roots of that plant extend to through this kernel that describes the root distribution, which we often take to be Gaussian. And likewise, the water uptake at a given point X depends on the plant at the point X prime, which the roots of that plant extend to the point X. Now this system of this model can be extended to describe a community of species with many biomass variables and the depth dimension. And importantly, it simplifies quite a lot when we apply it into specific context. And so basically, this is the platform of models. Let me show you solutions, typical solutions of this model, this type of model in one space dimensions. So we describe them by classification diagrams where the vertical axis is some state variable like the biomass. And it's a pattern, so we take, for example, the spatial average or L2 norm, and that doesn't matter much. And the horizontal axis is the bifurcation parameter, in this case, precipitation rate. And what we see here, and the convention, for those who are not familiar with bifurcation diagrams, solid lines represent stable state, and dashed lines unstable state. So we have two uniform states here, in those in black lines, the Bersol solution at zero biomass. It is stable up to some precipitation, beyond which it becomes unstable, that is, it can germinate and grow to form vegetation. It tends to form uniform vegetation, but uniform vegetation is stable only at high precipitation. As precipitation drops from high values, we hit a critical precipitation value where vegetation patterns develop, and one can do linear stability analysis of the uniform vegetation state and obtain dispersion curves, that is, a curve of showing the growth rate as a function of the wave number of the propagation of the uniform state. And what we see indeed, that at P equal PT, P for P-Turing, all modes decay, their growth rate are negative, except one, which is with wave number Kc, which doesn't decay, and as P drops below Pt, we have a band of modes that grow. Another way to analyze this pattern formation phenomenon is to calculate the so-called booster balloon. Those patterns, those modes or perturbations within this band of wave numbers grow and develop into periodic patterns with the same wave number, but only a subset of those patterns is stable. And those that are stable are within the booster balloon, so within this range here. Okay? And booster balloons have been observed in nature, in vegetation patterns. You see here the same environment, banded vegetation or striped vegetation with different wavelengths. An important point to stress here is that periodic patterns persist to significantly lower precipitation values in comparison to uniform vegetation. And this is because plants in periodic patterns can draw water from the versal surrounding, and this is an extra source of water in addition to the drier rainfall, and this is why vegetation patterns persist to lower precipitation. In two dimensions, sorry. In two dimensions, and during its stability, we have this critical mode that begins to grow, but if the system is isotropic, those modes can be in any direction. So the strike modes can be in any direction, and actually all modes in all directions begin to grow simultaneously. But triads of modes that are 120 degrees apart resonate with one another, and for that reason, they amplify one another and go faster than other modes. And that leads to hexagonal pattern, which is basically the superpositions of these stripe modes. So at the during stability, we first get hexagonal gap pattern, and as precipitation decreases, stripe patterns become stable, and at the lower precipitation, hexagonal spot patterns become stable. And let me show you now a simulation done by a former student, maybe it's here, I don't know, where we start from high precipitation, where the vegetation is uniform, and we slowly decrease the precipitation rate. And as you see, hexagonal gap pattern develops, and as precipitation further decreases, those gaps merge together to form a stripe segment that eventually lead to, as you see, stripe patterns, labyrinth-like stripe patterns. And as precipitation further decreases, at some point those vegetation stripes will break into spots, and these morphological changes from gap patterns to stripe and stone to spots increase the versal areas around vegetation patches, and thus the additional water resource. And again, also in two dimensions, these morphological changes lead to patterns that can persist to lower precipitation values. And these three basic morphologies are widely observed in nature. And here you see the hexagon spot pattern, which eventually will disappear in the collapse of the soil. So current theory appears pretty successful, and actually there are many more successful tests, but there are some observations that challenge it. And this brings me to the interplay between vegetation patterning and phenotypic transitions. And here I want to refer to a fascinating phenomenon, the so-called Namibian fairy circles. These are huge areas of grasslands with circles of bare soil, like the one I showed you in Australia. This is in Namibia, and these are the first to be observed. Here, the diameter on average is 6 meters. And this later has been found in Australia by Stefan Getzen in collaboration with our group. And recently, remote sensing study patterns of this form, 30-circle, 40-circle-like patterns, have been found in 263 sites. So this appears to be a wide, well-observed behavior phenomenon. Now, there are two main hypotheses for the mechanism that form these fur circles. The one is pre-existing nests of termites that feed on the grass roots, and then the circles of the termites form the pattern, the hexagonal patterns of nests, and the circles are basically the foraging area of these nests. The second hypothesis is water vegetation feedback that induces tuning stability, as we discussed already. This is a highly debated topic, and I don't want to go into that. I want to focus simply on the simpler second hypothesis. The second hypothesis is simpler because the system is obviously water limited, rainfall between 10 to 250 millimeters per year. So water and vegetation are two essential elements. And if we can explain everything, our observation, just with the water vegetation feedback, without resulting to a third agent termite, then at least it's simpler and nicer. Now, I was mentioning the water transfer mechanisms, overland water flow, and water conduction by laterally spread roots. Both of them do not apply to Namibia because the soil is sandy, meaning infiltration rate into the soil are high, and water infiltrates before it has the chance to flow over land. And the roots are pretty confined. The lateral extent is a few tens of centimeters. This cannot explain the few meter scale of the first set of patterns. So the remaining water transport mechanism is saltwater diffusion. And now, as the classical theory predicts that gap patterns will first appear as precipitation drops down, which can explain for the first circle, those gaps can be interpreted as first circles, but the classical theory predicts that at lower precipitation, we should see also stripes and spot patterns, but these are not seen despite the fact that there are regions and times of very low precipitation. What is observed instead is multi-scale patterns where as precipitation drops, the metrics, vegetation metrics in between the circles, which is pretty uniform at high precipitation, becomes pattern, forming small-scale spot pattern at low precipitation. So what we miss here? Well, the current theory overlooks a robust response form to aridity, which is phenotypic changes, as we discussed earlier. So let's try to incorporate it and see whether we can explain those puzzles, basically absence of stripe and spot patterns and the appearance of multi-scale patterns. Now, soils are generally layered. In Namibia, we have a topsoil, which loose sandy topsoil, and compacted deep soil below it. And the deep soil is moister because the evaporation rate in topsoil is much higher than the deep soil, and these water content differences within the first circle at different depths is these measurements demonstrated. Low in the topsoil, high in the deep soil. So this suggests hydrotropism as the phenotypic change. If the deep soil is wetter, we may expect a change, a phenotypic change from shallow-rooted plants to deep-rooted plants. Okay, but the deep soil is more compact. So the deep-root plants experience more compact soil where the hydraulic connectivity or soil diffusion is much slower, and this actually will give rise to the short scales that we are looking for. So we extended the model to include the two phenotypes of V1, the biomass of short-rooted plants, V2 of deep-rooted plants, W1 and W1 or the related variable S1 represent the soil water content in the topsoil, and W2 and S2 in the deep soil. I won't go into the details of this model, but I'll say that the phenotypic transition is described by these terms, where FS is the phenotypic transition rate, which is positive if the soil water content in the deep soil is larger in the topsoil and zero otherwise, in the step function. And we have non-local dispersal without convolution. We have infiltration, percolation of water from the topsoil to the deep soil, and we have a large difference in the soil water diffusion, low in the topsoil and high in the deep soil. And when we study uniform solutions of that equation, especially uniform steady-state solutions, we find two versatile and mixed phenotype uniform vegetation. And when we do the linear stability analysis of the uniform mixed phenotype state, we see a dispersion relation showing two modes that can grow simultaneously, one with short wavelength and one with large wavelength. And when we look at the eigenvectors, we can easily associate the short scale with deep roots and the long scale mode with the shallow roots. So the short scale mode will describe the short scale spot pattern within the matrix, whereas this mode describes the long scale torus circle pattern. Now we can simulate the model equations and see whether we can reproduce empirical observations. So first, switching off phenotypic transitions, setting the rate to 0, we get a classical sequence morphological changes from gap pattern to stripe patterns to spot pattern. But when we introduce finite phenotypic transition rate, we can get still gap patterns, but then the dynamics switch to the short scale because phenotypic change to deep-rooted plants takes place, and the deep-rooted plants, as I said, experience the compact soil with the low hydraulic connectivity. So we get this short scale pattern and basically multi-scale pattern, and at yet lower precipitation, we get a ring pattern. So this is what the model simulations predict. And this is very much like what empirical observations show. And another important significant observation is that in the model, basically the model studies, is that if we look at the precipitation at which the spot pattern, the last spot before collapse of Earth soil, the precipitation at which it collapsed to bare soil. This is at 123 millimeter per year for the parameter set that we chose here. For the same parameter set, the collapse of these ring patterns, elastic patterns, in this case, occurs at 46 millimeter per year at much lower precipitation. So the combination of the two mechanisms, spatial separationalization into periodic patterns and phenotypic changes, results in significantly higher resilience. Now, with the model, we can study the effect of other rates of phenotypic transitions. For example, if we reduce the rate, that means that, and precipitation is decreasing, that means that we can get, the system can get to lower precipitation rate before transition to deep-rooted plants can take place, and therefore we can see the first morphological transition from hexagonal gap pattern to stripe pattern, and only then get to the multi-scale now stripe patterns. And if we further decrease the phenotypic transition rate, then the system can reach yet lower precipitation before the transition to deep-rooted plants take place. And then we can get a large-scale spot pattern before, again, we get multi-scale pattern, this time spot patterns. In contrast, if we increase the precipitation, the phenotypic transition rate, we can get transition to deep-rooted plants before uniform vegetation destabilizes, and then we get only the short-scale patterns, and this is observed in Namibia. Now, what I discussed so far is a model that is specifically geared to the specific Namibian pericycle ecosystem. But we would like to analyze the interplay between vegetation patterning and phenotypic changes more generally. So what we did here, we actually modified the model or extended to include what we may call an order parameter phi for phenotypic changes, like describing phase transition. And indeed, we chose the simplest form, normal form, or Ginsburg-Landau form, where the reaction part gives rise to a transition from one phenotype to another, from fast-growing phenotype to stress-tolerant phenotype. For example, by closing stomata, which reduce the loss of water to the atmosphere. This is the reaction part. The diffusion part of the phenotype is motivated by observations that the phenotype actually can diffuse, in the sense that stress plants can signal their stress and elicit phenotypic changes in nearby plants. And this is experiments done by Ariel Novoplansky and his co-workers in our institute, where he has a series of plots with plants that have roots in two adjacent spots. So this is a single plant having roots in two spots, and likewise for the other plants. And he subjected only the first pot to water stress. And what they observed is that after a certain time, so in response to this water stress, that plant started closing its stomata. There's the phenotypic change that took place. And then after some typical time scale, this plant here, which is not subject to water stress, also closed its stomata. And after a while, also this one, the third one, which has no direct relations to the first one, also closed its stomata. So we have here kind of signaling that is partly by the release of the hormone ADA, abscisic acid, by the roots of the stress plants, but then the unstressed plants, they absorb this hormone ADA and interpret it as an indication to stop, to close stomata, and this is to produce more ADA that excrete out to the soil and the next plant absorber and so on. Okay, so this is work in progress, and I don't want to go into details. Let me just show that a Basquesh diagram of uniform solutions, showing that phenotype 1, the fast-growing species phenotype, is stable at high precipitation, but loses stability to the stress-tolerant one as precipitation is decreasing. And the stress-tolerant indeed persists to lower precipitation rate compared to the fast-growing one. And this bifurcation diagram is very similar to the same topological structure, stem structure than the vegetation bifurcation diagram that we've seen before, except that here, this is a mechanism at the level of a single plane, the organism level that produces a stress-tolerant phenotype. And here, it's a mechanism at the population level of many plants and spatial patterning. And this is the kind of stress-tolerant behavior at the population or state of the population that allows vegetation patterns that allows it to persist lower precipitation levels. And the next step is to study the interplay between these two mechanisms. And let me just note here that this may be relevant to other contexts. For example, bacteria that are known on one hand to form spatial patterns, and on the other hand, to go through phenotypic changes to phenotype that can tolerate antibiotics. So, and these are experiments by Nathalie Balaban, and so actually this is an important problem to study generally with applications to various research fields. Okay, with the time that, how much time do I have left?
Oh, maybe like 5 minutes.
Okay, I'll try.
5 or 8 minutes.
Yeah, okay, thank you. So let me go to the interplay between vegetation patterning and community reassembly. Reassembly that involves shifts from fast-growing species to stress-tolerant species. And here we use a trait-based approach, which means that we don't focus on different species, but rather on traits, more precisely on groups of species that share the same values of some small number of functional traits. This approach is getting more attention in ecology today because it's more geared to address functional questions like how a given system will tolerate a given stress. For that purpose, we need to know how the growth rate will be affected, how the tolerance to water stress will be affected. We are not interested in traits that distinguish between different species, but are not relevant for the stress. So with this approach, we focus on two functional traits, growth rate and tolerance to water stress. And we know that there is a trade-off between these two traits. So we can introduce a single trait parameter, a trade-off between the two, so that chi equals 0 corresponds to a fast-growing species and chi equals 1 to the extreme, other extreme edge, stress-tolerant species, but we can have all the species in between. And then I won't go into the extension of the model to describe, to account for it. But the community is described by a time-dependent biomass distribution in the composed physical space and thread space. And in that distribution has typically fast-shaped solutions in thread space. If there is also patterns in the real space, we take the spatial average here in this diagram. And the important point here is that so out of the whole species pool, which is the whole unit interval of chi, a subgroup, a community emerges because of competition and facilitation between different species. And that community, curve describes the community, gives us several community level properties. The first is the location along the chi axis, which we quantify by the chi value of the most abundant species, chi max. And this gives quantify the composition of the community. The width of this distribution is a measure for the functional diversity. The area behind the curve is the total biomass, and there might be cases where distribution is not even, and then we can study the evenness of the distribution. And those community level properties we can study now by varying, we can vary environmental parameters, for in particular precipitation, and study how these community level properties change. And this is a community bifurcation diagram that we obtained, where the vertical axis now is community composition. We have here the uniform vegetation community solution that is stable up to a critical precipitation value, and then there is a Turing bifurcation to periodic patterns, and these are solution branches of periodic patterns with increasing wavelengths as precipitation decreases. And what this bifurcation diagram tells us is the following. First, that while the community is spatially uniform, there is a shift from fast-growing species to stress-tolerant species, not surprising. But when the Turing bifurcation is hit and we have a transition to periodic patterns, then we have a shift back to fast-growing species, because now the surviving plants benefit from the extra source of water, the water they draw from their basal surroundings. Another observation is that the community hardly changes along this periodic solution branches. If we follow this blue solution curve, for which wave number is fixed, we see that chi hardly changes over a wide precipitation range. At some point, it starts increasing, so there's a shift to stress-tolerant species, high chi values. But then there are other solutions, periodic solutions with longer wavelengths and lower chi values. So this suggests that spatial patterning may buffer community composition changes, and direct simulations indeed, direct simulations indeed confirm that. What I show here is the community boosted balloon within which we have stable patterns, and superimposed on that boosted balloon, we plotted here trajectory of the response of the system to decreasing precipitation in time, starting from high precipitation, and the community composition now is given by this color bar. So blue is fast-growing species and red stress-tolerant one. And what we see is that there is a long or wide precipitation range where the community is dominated by fast-growing species. But as we approach, and this is where the wave number doesn't change, but as we approach the boundary of the boosted balloon, the shift to stress tolerance begins. And as we cross the booster balloon, then we reach an unstable periodic pattern. The system converges to the basin of production of longer wavelengths periodic patterns, low wave number. And this is accompanied by a transition back to fast-growing species to some extent. And that process repeats itself one more time, and then eventually the system collapses to bare soil. At some point, the spatial patterning cannot compensate for the reduction in rainfall. Stress-tolerant species develop up to the point that collapse to bare soil occurs. The key to understanding this buffering effect of the community change is the high spatial plasticity of vegetation patterns. And in one-dimensional system, there are two expressions of this spatial plasticity. The first is patch thinning along each solution branch. So let's focus on this point here. The patterns look like this, and this point here, the pattern looks like this. So the horizontal exited space. And what you see that since it's the same solution branch, same wave number, the number of patches have not changed, but they become thinner. They become thinner, there's more versos along in each patch, more water for the plants to take up from this surrounding. The other expansion of spatial plasticity is patch dilution by transitions to lower wave number patterns as the booster balloon is lost. And so let's compare this point with this pattern to this point with the transition to that point on the solution branch with lower. So now we have less patches, there's patch dimension or patch dilution. And again, the effect is that each patch has more versos surrounding it and more extra source of water. In the booster balloon, patch thinning appears as horizontal line and patch dilution is the vertical line. And as I said, both processes increase water availability and compensate for the reduced precipitation, thereby keeping water availability more or less constant and buffering community change. There's no driving force for community change to stress-tolerant species. Let me say just a few words about these complicated solution branches that snake back and forth. This phenomenon is called phenotypic snaking, and it's a pretty hot topic in the mathematical community. They describe localized patterns, like localized pattern domain in uniform vegetation, or otherwise localized uniform domain in otherwise pattern vegetation. And the important point from the ecological point of view is that this pattern, the uniform domains, provide different niches than the periodic pattern. In the uniform domain, there's more water stress. So here, stress-tolerant species reside. In the periodic domains, water stress is relaxed to some extent, and faster-growing species can reside. And this leads to higher functional diversity, and as compared with the periodic pattern. And actually, for different parameter range, this effect can be much more significant, leading to bimodal biomass distribution along the trait axis. I talked about the expression of spatial plasticity in one dimension. In two dimensions, we have the morphological changes, which are also lead to spatial plasticity. And with this, I will conclude. So vegetation pattern formation, which is the population level response to tolerate water stress by transition from gap pattern to stripe pattern to spot pattern. But what we saw is that there is a need, at least in some cases, to extend the theory to include phenotypic changes and community dynamics. incorporating phenotypic plasticity. We saw results outstanding first set of puzzles and reveals previously underestimated resilience. Thus, multi-scale patterns are significantly more resilient, and incorporating community dynamics unravel the buffering effect of vegetation patterning on community structure. The extended theory can highlight ways to evade the loss of ecosystem function by understanding alternative response pathways. I discussed here two such pathways, those multi-scale patterns and buffering of community structure. But there are many more pathways to consider, which brings me to this ERC synergy project, where the purpose of which is to study pathways of resilience and evasion of tipping in savanna and tundra ecosystem. And those of you who want to hear more about this project, I'll be happy to tell you. And let me conclude thanking my collaborators, my former postdocs, Jamie and Gidesh, and my current postdocs, Mitchell and Joydeep, and my colleagues, Hannes Super from the University of Oldenburg, Stefan Getzen from Göttingen University, and Helmut Zwack from the GU. And here are some relevant references. And thank you for your attention.
Thank you very much for this really nice talk on what Jürgen described as nonlinear physics of ecosystems, and you covered a lot of topics. And I request the audience to type their questions in the Q&A box, and in the meantime, the panelists, if they want to ask questions, they can ask directly also. So please go ahead and type your questions in the Q&A box. Go ahead, Professor Lakshman.
Yeah, I have some basic questions. So the model you described essentially applies to a particular region. I think it's, you said Namibia. So how does the water, the rain, the soil quality, etc., affects the model? Can this analysis be extended to other regions of the world?
Well, as I said, we basically have a platform of models, and when we applied it to the Namibian first cycle system, then we realized that some water transport mechanisms are not relevant, like overland water flow because of high infiltration and water conduction by lateral routes, and only saltwater diffusion is significant. And what is interesting in that particular system is that because of the inhomogeneity in the depth dimension, loose sandy soil in the top and compacted soil behind it, deeper, and then we have fast soil water diffusion in the lateral direction. If you think of a precipitation rate that exceeds the percolation of water from the topsoil to the deep soil, then and the fact then topsoil becomes saturated, hydraulic conductivities become very high, and it cannot go deeper, so it goes to laterally, and this explains the large scale of the first circles. Because usually,
so do you compare your results with the actual experimental situation?
This is a very good question. We have new samples of columns of soil depths taken from Namibia. This is ongoing research. And what we plan to do is to analyze them and get from them characteristics of the soil, put them use them in the hydros, which is a software based on the Richards equation to describe the saltwater dynamics, and see whether with these parameters we can explain the large scale of saltwater diffusion in the lateral direction. And this is my PhD student, Shachar, whom we saw at the beginning, is working on.
Okay. Okay, thanks.
Ehud, can you see the Q&A box? There's a question from Sangeeta. Or should I read it out? It's in the Q&A box. What kind of instabilities lead to transition from patch thickening to thinning? That's the question. It's from Sangeeta. I think she was.
Can you repeat? I didn't hear.
The question, you can, if you click on the Q&A box, you can see the question yourself. Can you
see chats in the chat?
Share, on the right side of share.
No.
Can you see the question?
No, I don't see them. But
okay, I will read it out, don't worry. The question is from Sangeeta Ujjwal. What kind of instabilities lead to transition from patch thickening to thinning?
Oh, patch thinning to?
Patch thickening to thinning.
Well, this is no, there's no instability involved. This is just following a given solution range, periodic solution range with a given wave number along the rainfall and along the precipitation exit. So decreasing precipitation is patch thinning, increasing precipitation along the solution range gives to patch thickening.
Thank you. If there are more questions, please type the questions on the Q&A box. Yeah, there is another question. Thanks for the nice talk. In the past, you also addressed plant self-organization patterns with regard to optimizing spatial restoration effects. Do you get new inspiration on addressing such restoration topics based on your latest work?
I believe you refer to restoration by water harvesting through periodic dikes. We know the answer is no. I mean, we have not studied further this or have not studied multi-level ecosystem response in the presence of these dikes that provide that harvest water to vegetation stripes planted along those dikes. So I cannot comment on the multi-level ecosystem response on that behavior. But this is an interesting question and an interesting topic to study in the future.
Okay. Are there more questions from the audience? Please type them on the Q&A box. I have a question myself. Do you see fractal patterns? And if so, can you model them?
Which patterns?
Fractal patterns of the form of a
fractal. No, and the theory does not predict them. The theory predicts. Well, we are talking not about single plants. We are talking about patterns of many plants, population of plants.
Right.
And then there is a given scale that is produced by the Turing instability. And then there are instabilities to longer wavelength patterns, but not to kind of scaling bands or having all scales like in fractals. So well, there is one limit where we studied that may give to scale-free patterns, patterns involving patches of very wide distributions, size distributions. This occurs according to our model studies when water transport is fast compared to water take-up or utilization. For example, if water diffusion is very fast compared to infiltration rate and water intake rates. Under this condition, we can get scale-free patterns. Not really scale-free patterns, but distributions involving kind of several scale, like 3-4 scale. There are observations. There are 2 nature papers in 2007 showing observations of scale-free patterns, but they didn't resolve possible heterogeneities that might have given rise to the different scales. So this remains an open question.
Okay, thank you. And I have one more question. I mean, all this is without any human intervention anywhere near that. But if there is, in reality, there are human beings and they do all kinds of things, is there any possibility of modeling that in some crude level?
Yeah, this is a very good question. And this is actually one of our goals to study. In this ERC project, basically what we want is to understand alternative best way of ecosystem response that give rise to resilience. So if the system is on a track to collapse, to a tipping point and collapse, and we know that there is an alternative pathway that is resilient, if we understand it and the difference between the 2 pathways, we may get ideas about how to intervene in the one pathway that leads to collapse in order to shift it to the resilient pathways. And we have already some studies, some results that suggest intervention of that kind. For example, and those interventions can, in general, be integrated with services that we extract from ecosystems. For example, an important ecosystem service is feeding livestock by clear cutting of fodder or by grazing. And what we found is that if we manage grazing or this clear cutting in a non-uniform way, we can prevent the system from collapse and forming patterns which are much more resilient. And actually, after finding this with model studies, we learned that there is, in fact, such a practice which is called rotational grazing, where grazing is not done uniformly, but they divide the area into sectors and proceed from one sector to another one and let the original sector regrow before coming back to it. And this is non-uniform grazing that now we understand that can increase the resilience of the system. So yes, human intervention in particular, informed intervention to increase the functioning of the system is something that we are studying and plan to study furthermore.
Thank you. There is one question in the Q&A box from Joshin John. Does these models account for the different timescales involved in the system? In desert, is plant growth happening immediately in response to the precipitation?
So then this is a good question, and there are many timescales involved in the response. And depending on the level of organization, phenotypic changes in the level of a single plant can be slow, but can be also be very fast. It depends on how much cost or effort the plant invests. So, for example, closure of stomata can be done in timescales of minutes to avoid water loss, and this is fast phenotypic change. But for example, there are phenotypic changes in reports, phenotypic changes from C3 plants to chem plants, or from C3 to C4 plants, for example, which involves different anatomical changes. So these phenotypic changes, which involve not only just biochemical reactions, but also structural changes, anatomical changes, they involve much more cost for the plant, and they involve longer timescales. This is the level of a single plant. Then there is the level of population forming patterns. Here again, we have different timescales depending on the species. So these are herbaceous species like annuals. Timescales can be pretty short, as of weeks. But if we talk about woody vegetation, trees, it can be years and tens of years. And then there are evolutionary timescales, which can be even longer. So there are many timescales, and this helps us simplify this overwhelming complex system because of this timescale population.
Okay, thank you. I think we answered all the questions. So I want to thank you one more time for accepting this invitation and give this wonderful talk. The next talk will be by Professor Tiago Pereira from the University of Sao Paulo on October 28th, at the same time. Of course, different parts of the world, the talk is on different time. So I'll send you a link with the clear timing. Thank you very much once again. Bye, everybody.
Thank you all for the good questions.